Hi, I'm Luke Yin, a senior at the Illinois Mathematics and Science Academy, and today I'll be presenting my project titled Efficacy of Tamivosertib, a MINC inhibitor in mitigating RDEB mouse pain. First, for some context, epidermolysis bullosa, also known as EB, is a class of rare genetic skin disorders characterized by extreme blistering of the skin. There are four main types, but for my project, I'll be investigating recessive dystrophic epidermolysis bullosa, also known as RDEB. This is one of the more severe forms of EB, and it's caused by mutations in the collagen 7A1 gene. The collagen 7A1 gene produces collagen 7, which creates the anchoring fibrils that connect the different layers of the skin. So therefore, in the absence of collagen 7, there's a lot of blistering that occurs because the different layers of the skin rub up against one another, which creates that blistering that we see in patients. So in the diagram up here, we can see that in healthy skin, the anchoring fibrils indicated by the purple ropes are all present, which means that the skin is connected. And then in the RDEB skin on the right, we can see that those anchoring fibrils are not present, which is why the skin is inflamed and blistered. And then here is what RDEB looks like in patients. So here you have the hand of an 8-year-old girl and the foot of a teenager, and you can see that there are a lot of mitten deformities and blistering throughout. Now for pain management and RDEB, pain is often very severe in RDEB and it's chronic. So these patients are often subjected to opioid treatments. However, the problem with opioids is that they have a myriad of side effects and they're not sustainable long term. Also, they are very dangerous when used in pediatric patients, and a lot of the patients with RDEB are children, which is why it can't be used. So therefore, developing targeted therapies for RDEB pain is very important. And for the purposes of our study, in order to investigate pain, we use a collagen 7A1 mouse model because it recapitulates a lot of the key characteristics seen in human RDEB, such as the blistering and mitten deformities that I showed earlier, which makes it an ideal model to study pain behavior and develop targeted therapies. In the diagram here, you can see that the wild type pot is very healthy and large, and there's no mitten deformities present. But in the mouse model, we can see that the pot is shrunken and that there are mitten deformities, just like we see in the human RDEB patient right here. And then pathophysiologically, it's also the same. Now, regarding RDEB mouse behavior, this on the left is a behavior known as nibbling, and it is indicative of pain. On the right, that's a behavior known as wiping, and it's also indicative of pain. So we know that these RDEB mice experience pain and itch in the same way that humans do. Now, my study investigates the MINC signaling pathway, which is one of the pain pathways involved in RDEB. Essentially, how it works is MINC phosphorylates EIF4E, and then that drives the production of pain-related proteins such as BDNF, which translates downstream into pain. So therefore, the hypothesis of my study is that targeting the MINC pathway with tamivosertib, a MINC inhibitor, will decrease pain in RDEB mice by reducing pain-associated gene expression in sensory nerves. Now, to actually measure the levels of gene expression pre and post treatment, we use what's known as RT-qPCR, which stands for Reverse Transcription Quantitative Polymerase Chain Reaction. This measures the level of mRNA in a gene, and that's important because when a gene is expressed, it produces mRNA. So by assessing the relative levels of mRNA pre and post treatment, we know if our treatment is effective. And in our case, we're looking at genes such as BDNF and EIF4E, which are directly involved in the MINC pathway. Now, our preliminary findings have revealed that the MINC signaling pathway is upregulated in RDEB. As you can see in the charts on the left, in the dorsal root ganglia, the x-axis shows the MINC-associated genes, the y-axis shows the fold change in mRNA, which is basically the level of gene expression, and then the gray bars show the wild-type mice, while the purple bars symbolize the RDEB mice. And in the dorsal root ganglia, there were significant increases in the levels of gene expression for all of the MINC-associated genes, and then that trend is also true in the front paws. On the right here, in the immunofluorescence stains, we can see that there is significantly more red in the RDEB stain. And the red here symbolizes phosphorylated EIF4E, which is one of the genes I talked about earlier that is involved in the MYC signaling pathway. Now for the results themselves, we found that the MYC inhibitor did reduce downstream MYC signaling in RDEB mice. So the left graph here, or the left chart here, shows the dorsal root ganglia, and then again the x-axis shows the MYC-associated genes, the y-axis shows the levels of mRNA. The yellow bars symbolize the control group, which is the RDEB mice treated with dimethyl sulfoxide, and then the purple bars show the RDEB mice treated with the MYC inhibitor tamivosertib. So that's our experimental group. And then we can see that for BDNF, MYC itself, and EIF4E, there were significantly decreased levels of gene expression compared to the control group. And then in the front pause, that's true for MYC and EIF4E as well. Regarding pain behaviors, which is basically the videos I showed earlier, here are some of the categories we assess. So orbital tightening, nose bulge, cheek bulge, etc. And we rate those on a scale of 0 to 2, with 0 being not present and then 2 being obviously present. So the x-axis here shows the behaviors at different week checkpoints, so week 1 and week 2. And then the y-axis shows the prevalence of the pain behaviors. And then again, the yellow bars are the control group, while the purple bars are the experimental group. So for pain, we can see that it took 2 weeks, but the pain was significantly decreased in week 2. And then for grimace, which is a pain behavior, it was significantly decreased in week 1. And then interestingly, for itch, there were no significant correlations, which makes sense because itch is exclusively, or MYC is exclusively a pain pathway, so it's uninvolved in itch, which means this is a good sign. Now, since we had such promising results with the MYC pathway, we wanted to see how it affected the NMDA pathway as well, which is another pathway involved in pain. How it works is glutamate is released onto the NMDA receptors here, and then it binds to it. And then the genes here, such as GRIN1 and GRIN2B, help translate that downstream into pain. So here are the results. We found that in the x-axis shows the NMDA associated genes, and then everything else is the same. So we found that for GRIN2B, there was a significantly decreased level of gene expression in our experimental group compared to the control group. And now for the conclusions. We now know that, at least in a novel approach, mice with a collagen 7A1 gene mutation are being used to study the itch and pain behaviors associated with RDEB, and that these RDEB mice do experience significant increases in itch and pain compared to wild-type mice, which is what we see in RDEB humans as well. And we also know that MYC, EIF4E, BDNF signaling may contribute to RDEB pain, and that pain is reduced by tamivosertib-induced MYC pathway inhibition. We also know that MYC inhibition may decrease pain in part by reducing NMDA pathway activation. So our preliminary findings identify a basis for pain in RDEB, and these results in RDEB mice indicate promising new directions for medical development, specifically targeting pain signaling pathways to manage pain not only in RDEB, but in EB as a whole. And I would like to thank these individuals at the Northwestern University Feinberg School of Medicine Department of Dermatology and the Illinois Mathematics and Science Academy Student Inquiry and Research Department for their assistance with the research process. Here are my references, and thank you so much.